Hello, today we'll talk about infrared spectroscopies. This is one of the widely used methods of physical methods of investigation. Um, it mainly help us to identify the presence and quantify functional groups, different functional groups that are affected by infrared. And based on that, the infrared spectrum can also be used for different purposes like comparison of various samples with standards and some other approach like that. Okay, here we go. IR spectroscopy studies how molecules absorb infrared light. It is mainly used to identify functional groups in organic and biological molecules, as I just reminded. In infrared spectroscopy, a sample is irradiated with infrared light and the transmitted or reflected light is measured, allowing structural analysis and quantification. Absorbance analysis involves measuring the amount of absorption of light by the molecules in a sample, which is wavelength dependent. Let me remind you that in UV spectroscopy, electrons jump from the ground state to an excited state, so that the absorption of various bands of UV or visible spectrum uh, occurs, and based on the shorter wavelength of this UV spectrum, the jumping position of electrons from the ground state to excited state differ, which means in red, okay, they might jump to closer. In violet or ultraviolet, they can jump to longer excited state compared to the nucleus. Okay, differing from this, okay, infrared irradiation cause molecular vibrations in different samples in molecules. So in the case of ultraviolet and visible light from differing from 0.2 to 0.78 micrometer or from 200 to 780 nanometer, the absorption occurs due to electronic transitions in molecules. In contrast, infrared light, which differ from 2.5 to 25 micrometer or from 2,500 to 25,000 nanometers, is absorbed due to the vibration and rotation of molecules, and the energy involved here in this case is smaller. I have to remind that that the treatment of different kind of compounds or the surface of different matter by infrared irradiation can reach higher temperature because it causes the molecules on the surface of the matter to be vibrated, which the energy that's absorbed cause the higher heat energy. But in this case, they cause electrons to jump from here to here, from the ground state to the excited state. It does not mean that the UV or visible spectrum have lower energy. They actually have higher energy, but that photon energy cause these electrons to jump. And here, the energy, photon energy, is low. Even if the photon energy is lower, the ultra infrared light causes the molecules of the surface of the matter to be vibrated more, so it turns to heat energy more. This is the difference that we have to keep in mind. Okay, and here we can find out about the instrumentation of the infrared spectroscopy. The main sources are the source of the infrared light, monochromator or interferometer. Interferometer is used for Fourier transform infrared, about which we are going to talk a bit at the end of the lesson. And there is a sample holder, and there is a detector in it, and computer processing is the last tool that is provided to all, let's say, tools studying different physical methods of investigation. I'd like you to pay attention to this second figure given here. The source is providing with the infrared light, and the reference compound is studied, and the infrared spectrum of the reference compound is obtained, and the samples that we are going to study is place it, and infrared spectrum of it is studied. So based on the differences in the infrared spectrum, we can find out some differences, or we can suggest some differences between these two samples, the reference and the studied sample. And this is a kind of model comparison. Here we can see this kind of infrared spectrum of various compounds can be obtained. We are going to talk about these things during the whole lecture. And this is about the unit 4,000 to 400 reciprocal centimeter, and infrared spectrum is divided into near infrared, mid infrared, and far infrared ranges. Here we can see that near infrared includes 14 to 4,000 reciprocal centimeter unit, and mid infrared includes ranges, let's say, from 4,000 to 400 reciprocal centimeter. This spectrum is widely used and considered to be most important. And far infrared includes from 400 to 10 reciprocal centimeter. And why do we use this kind of reciprocal centimeters? This is easier to link the frequency with the energy, let's say. Here we can see that the wavelength of the infrared differs from 0.75 to 200 micrometers. In nanometers, it is from 750, 770 to 200,000 nanometers. And here we can see in micrometers, it is from 0.75 to 200 micrometers. But this is about the wavelength. But in infrared spectroscopy, we use the units like from 13,300 to 50 or 10 is given here with the unit reciprocal centimeter. Why is it like that? Because it's easier to link again with the wavelength, to link the wavelength with the energy. The standard unit in IR spectroscopy is the wave number expressed as reciprocal centimeters. Yeah. And while most of us used to thinking of light in terms of wavelength, like nanometers, infrared spectroscopy uses wave numbers because they are directly proportional to energy. The smaller the wavelength, the higher the energy, the higher the frequency, the higher the energy. That's why, okay, the higher the energy, the higher the wavelength is used for, okay, this kind of, let's say, unit. Let me remind you that these pointers are used to describe the wavelength, and these points are used to describe the frequency of the number. Okay. The meaning of 14,000 to 4,000 reciprocal centimeter stands for the numbers of these cycles, which means in 1 centimeter length, okay, there are 14,000 to 4,000 this kind of cycles, repeated cycles of the wavelength. Okay, this is the point that we have to keep in mind. And another point we have to keep in mind is that for the sound, yeah, we use hertz, for which, okay, the unit includes the second, number of cycles per second, and here number of cycles of the wavelength per centimeter. Here the unit is length, and in hertz the unit is time. Okay, by these two things they also differ. And let me remind, this is amplitude, and this is wavelength, and this is the cycle, number of cycles. Okay, we can claim they are the same, but they differ from each other with this 0.25 unit, let's say. Okay, so since 14,000 to 4,000 reciprocal centimeter is closer to the photon energy that the different wavelengths own. This kind of units are used for IR spectrum. Let's go. And the mathematical definition can be described with this. Mu equals 1 divided by the wavelength, and the energy can be expressed with this. Energy equals Planck constant multiplied by speed multiplied by the frequency, yeah, which is described, which equals 1 over the wavelength. And the wave number is the frequency, and this frequency unit is length, centimeter, not time, second. Okay, we talked about this now, and next, and how to interpret the scale. When you look at an IR spectrum, the scale usually runs from high to low, from left to right. You can think of it like a guitar string. High wave numbers like from 2,500 to 4,000 reciprocal centimeter, actually from 4,000 to 2,500. This represent light, fast vibrating bonds like the bond between oxygen and hydrogen, or between nitrogen and hydrogen, and they take a lot of energy to move these light atoms, because the number of frequency is higher compared to this one. And mid-wave numbers range from 2,500 to 1,500 reciprocal centimeter. This is where you find the triple bonds or double bonds. And low wave numbers from 1,500 to 400, let's say, reciprocal centimeters, are termed fingerprint region. The fingerprint region is used for identifying, let's say, low energy vibration of the entire molecular skeleton, or some bigger regions of molecules consisted of several functional groups. So these are the pointers we have to understand while we are analyzing the IR spectrum of different compounds. And this is about the quick conversion tip. Okay, again, UV spectroscopy is based on the jump of the electrons from the ground state to the excited state. And IR spectrum is based on the vibrations of the molecules. Okay, here we can see two different types of vibration: symmetric stretching vibration, IR inactive vibration, or antisymmetric stretching vibration in two different molecules, carbon dioxide and in water. Here we can see symmetric stretching of IR inactive vibration. It is inactive in this condition, and antisymmetric stretching vibration has 2,349 reciprocal centimeter unit for carbon dioxide. It can differ. It can differ from the symmetric stretching like this, that one oxygen atom is located at far distance from the central atom carbon, compared to the distance between carbon and another oxygen atom in the molecule. Okay, this is about the stretching. It's not about bending. That's why here we can see the linearity of the molecule is saved, kept, and the grade of between two hydrogen atoms in water molecule is kept as well. So this is about stretching, not bending. Here we can see that symmetric stretching vibration of water has absorption at 3,552 reciprocal centimeter, and anti-symmetric stretching vibration has a absorption at 2,349 reciprocal centimeter, which can be described like this. So in antisymmetric stretching conditions, one atom is located to a far distance compared to another same atom of the molecule compared to the distance related to the central atom. Okay, so these are the differences between symmetric stretching and asymmetric stretching in examples of carbon dioxide and water molecules. Okay, besides symmetric or asymmetric stretching, there are bending differences, as I reminded in the previous page. So they can differ from each other by different positions, like terms like scissoring, rocking, wagging, twisting. Here we can see symmetric stretching and asymmetric stretching, as we witnessed in the previous page. And these are the pointers that could be claimed as bending. These two atoms, for example, the same atoms can come closer to each other and cause the scissoring, let's say, bending shape. All these kind of differences cause the formation of rocking bending. Okay, so these are the pointers we have to understand, and stretching vibration has this kind of differences. Okay, these two atoms have this kind of distance, and while they are stretching from each other, the distance can be far. So that is the reason why vibration is how vibration is described, and this describes the bend. Okay, two different atoms might come to closer distance, and here in stretching they are in far distance uh, compared to, let's say, normal non-excited condition, and in bending condition they come closer to each other. That could be described like this. Okay, so these are the differences between stretching and bending vibrations. In bending, the atoms are closer to each other. In stretching, they are far from each other. Okay, and another point that we have to take and keep in mind, we have to take into account and keep in mind, is that the vibration might cause a change in dipole moment. I'm not going to talk about dipole moment. That if you are interested, try to link dipole moment change with the vibration as well. Okay, here we can see simple layouts of the vibrational modes associated with the molecular dipole moment change that are detectable in an IR absorption spectrum. In addition to the two stretching modes, the four different bending vibrations are shown. We talked about these four different things. This is symmetrical stretching, as we talked. They can be stretched into this directions. And here we see asymmetric stretching. One atom is stretched, another atom might be located to a closer distance. And in wagging, here we can see this kind of changes. In rocking, okay, these are about the bending, yeah. And here you can see rocking. This is twisting and scissoring. As we talked, they might come closer to each other, or they can then go to the same distance compared to the location of rocking, or this kind of distance in a condition in terms of wagging. So based on this kind of changes, let's say the vibrations are formed, and those vibrations cause the absorption of some bands of infrared radiation. And here we can understand how we can interpret the signals and the spectrum. Let's say when we have the IR spectrum, we can obtain this kind of spectrum, and what this absorption and transmission stand for. Here we can see no IR light for particular wavelength absorbed by sample. Here the sample is not absorbing infrared, and that not absorbed infrared line might show this kind of, let's say, spectrum with high level of transmission. Okay, this is transmission level, this is absorption level. If some light is absorbed, okay, it goes down. If not, okay, transmission level is high, so the peak of the transmission standards, is located here. Okay, if some light spectrum is absorbed as indicated here, yeah, so then we might have this kind of absorbance. Okay, this is high level of absorbance and very low level of transmittance. Percentage of IR light transmitted through the sample and detected is very low. Low level of transmittance and high level of absorbance, low level of absorbance and high level of transmission. Okay, these are the points that we have to understand. Sometimes we might have absorbance peaks that could be indicated like this. Yes, not this kind of from upside down, but in that case from down upside case. Okay, next. When we obtain the IR spectrum of different compounds, we might have something like this. This is the IR spectrum of vanillin that has a structure like this, where we can find out aldehyde group, hydroxyl group, or oxymethyl group, something like that. So it has many functional groups, OH. Yeah. Methyl group, OH group, yeah, carbonyl group, carboxyl group, CH group, C double bond, H group, yeah, CC double bonds, yeah, many different bonds. That's why we might have this kind of sharp structure. This kind of spectrum with sharp, let's say, absorbance levels. Okay. Now we are going to understand how we can interpret the IR spectrum. A typical mid IR transmission spectrum showing schematically the typical absorption lines associated with the vibrational modus of molecules, organic compounds, and other contributions of biological and non-biological nature. Okay, again, if the uh, absorption levels belong to this kind of spectrum, so then we can claim that the compound has OH groups and H groups or CH groups. Yeah, if the absorbance belongs to, let's say, the spectrum between 2000 to 2500, so we can claim we can claim the presence of compounds holding carbon-carbon triple bonds or carbon-nitrogen triple bonds. If the absorbance is located between 1000 to 2000, yeah, from 2000 to 1500 actually, reciprocal centimeter, so high probably there we have carbon oxygen double bonds, carbon carbon double bonds, carbon nitrogen double bonds. Yeah, okay, and this absorbance range belongs to skeletal vibrations and single bonds, which means, okay, this carbon oxygen double bond or carbon nitrogen double bond or carbon carbon bonds belong to carbon oxygen, carbon nitrogen, carbon carbon double bonds that are affected by other neighboring functional groups. Okay, which means they are present here, but in the composition of more complicated structure. That's why it is interpreted as fingerprint region, it is used for skeletal vibrations and single bonds. And here we can see similar results from 500 to 500, 1500 is indicated here. It's not shown here in this example, but here we can see carbon-oxygen double bonds. Yeah, carbon-oxygen double bonds between 1500 to 2000. Yeah, here, or triple bonds between carbon-nitrogen or carbon-carbon between 2000 and 2500 reciprocal centimeters is indicated here, right? And here we can see more informative data that OH stretches, yeah, or nitrogen NH stretches, or CH stretches belonging to alkyne group, yeah, or alkenyl group, or CH stretches belonging to alkyl group, different groups. That's why, okay, the spectrum is, let's say, quite complicated in order to obtain some exact results, to claim the presence of some functional groups. We have to carry out kind of thorough comparative analysis of functional groups with those that were established in different compounds. That is why, let's say, that's why in this example here we are reminded the comparison of the sample with the reference compound's IR spectrum. Okay. Otherwise, identifying the presence of functional groups can be quite difficult for the new learner. And here we can see frequency absorbance in different reciprocal centimeters belonging to different functional groups. For example, for CH, okay, these are the functional groups of the butanol. I have to remind this belongs to butanol and this belongs to butanol as well. Okay, the data are obtained from this website, and CH has 2,650 to 3,310 reciprocal centimeter. Yeah, somewhere here, right? So this absorbance might belong, or we can claim belong to CH groups or some other groups that could have other interactions. For example, OH group or OH group in alcohols, OH group in acids, something like that. Okay, CC bonds can be detected here from 720 to 1175. Yeah, CO bonds from 1120, 1310. Yeah, carbon-carbon double bonds somewhere here, 1600 to 1690. Yeah, somewhere here, and carbonyl bond 1630 to 1850. Yeah. Another point we have to keep in mind is some absorbance might have sharp forms like this, yeah, or broad forms as is indicated here. These are other points we have to take into account while we are analyzing the IR spectrum. And now we are going to see some exact explanations in exact functional groups. Here we can see the peak belonging to the carbonyl group. Strong, yeah, the strong. Here we can see it is strong and it is sharp, and the frequency is between 1750 to 1600 and 80 reciprocal centimeter. This is the carbonyl group in acetone. And here we can see two different absorbance peaks belonging to carbon oxygen bonds as indicated here, yeah, with the red colors, or oxygen hydrogen bonds as indicated this with this blue color. Here we can see it is broad for oxygen and hydrogen in this case, yeah, from 3,200 to 3,550, yeah, between as indicated here between 3,200 and 3,550, and the OH 3,200. I'm sorry, this is CO, not OH. I'm sorry, it's actually wrong. Yeah, OH is here. This belongs to 3,000. This one, I had to correct it. Okay, OH blue color is here actually. OH blue color 1,000. Okay, this is about OH. I'm sorry. Yeah, it is for OH. It is correct. Yeah, it is OH. But the second one is CO. This is not OH. This is about the CO from 1,787 to 1,205. Yeah, this is about CO, not OH. CO. Okay, the R is human. I am a human. Actually, I copied them from this website, but they had a technical mistake. I had to prove it. I had to correct it. Okay, and another one. Here we can see. Here we can see another example in carboxylic acid group.